Maybe before we jump into new stuff today, which was lipids, let's take a second to review carbohydrates and those other two lipids, and then we'll start new stuff. And I'm going to grab some colors and make my way over here. We're going to need to know the 4 different biomolecules. So I'll say biomolecule. And each biomolecule we're going to have to know in terms of their individual unit, what we would call a monomer. And then when we stack 3 or more of these monomers together, we get a polymer. We saw examples of monomers of carbohydrates and polymers of carbohydrates, and we went through 3 polymers of lipids. So let's review those carbohydrates. If the biomolecule in question, I'll just give myself some parameters here. Maybe that's enough space. If the biomolecule in question is going to be carbohydrates, we listed at least 4 examples of monomers of a carbohydrate. All of these monomers fell under the category of what we would call monosaccharides. Mono, we said, was 1. Saccharides are sugars. Can anybody remember what some of those monosaccharides were? Glucose was 1. Yeah, we said glucose had one of those hexagon shapes. I heard galactose. It also had a hexagon shape, but the functional group was in a different location between those two. I heard fructose, I believe. Fructose was in a pentagon shape. And then ribose. It too was in this pentagon shape. So we had some examples of monosaccharides, and then we looked at 3 examples. Let me take that back. There were 4 examples of polymers of carbohydrates. 3 of those 4, we had to recognize their structures. So carbohydrate polymers were polysaccharides. And without going backwards in the slides, let's list some of those polysaccharides. Yes, and let me maybe color coordinate these polysaccharides. There were 2 that we saw in plants. I'll try and write those in green. And then 2 others that we're going to find in animals. Maybe I'll save that for a second. But I heard somebody say starch. So starch, we drew that just a second ago. It looked like a coiled spring. We'll label this as starch. And we said all along that spring were individual glucose molecules. So starch, we said, was easily digestible as an enzyme. You could just make a single snip and release one of these glucose molecules into the bloodstream. So what was the function of starch? How did plants use starch? Yeah, it's how they stored their energy in the short term. So short-term energy. So short-term energy storage in plants. If that wasn't clear with the green color. But there was another polysaccharide in plants, and what was that? That was cellulose. And we can recognize cellulose as being different from starch because cellulose kind of looked like a lasagna where there were multiple layers. And each layer was a chain of glucose molecules. So each of these glucose molecules are covalently bonded together by this green line. And then in addition to the covalent bonds, there were hydrogen bonds locking the different levels together. So there was an entire lattice work of covalent bonds and hydrogen bonds that locked the cellulose molecules together. So as an enzyme, you could come in here and make a SNP, but you haven't released any glucose molecules. This is not going to be good for storing energy, but it would be a good cell walls in plants. So cellulose functions as the main material in cell walls. And then that takes us to the polysaccharides in animals. When we look at animals, there was really only one polysaccharide that we had to recognize the structure of, and it looked something like this, like a branching tree. Yeah, this was glycogen. We said glycogen was in a branching structure, and each branch was just a chain of glucose molecules. So due to its structure, it was also easily digestible. You could make a single SNP as an enzyme and release a glucose molecule into the bloodstream. So glycogen is how animals stored their short-term energy. So short-term energy in animals. Don't have a lot of room to write with this one, but hopefully you can read that. Glycogen is short-term energy in animals. And then there's only one other polysaccharide in animals. We don't have to recognize the structure, but we do have to know that the other polysaccharide is called chitin. Let's maybe put a little distinction between this. When we're thinking about chitin, we said it was found in fungi cell walls and also the exoskeletons of insects. That's right. So fungi cell walls and exoskeletons of insects. All right. Sadly, I'm going to have to erase that so that we can make room for the monomers and polymers of lipids. But we're going to do each of these little columns for the 4 biomolecules, as this will be helpful for the exam. I'll give you a second. Welcome to take a picture of it if you want. Let me know when we're good. Who knows, you might have to recognize those structures. You might have to give examples of polymers. Maybe I'm describing the functions and you have to pick out which one of those we're talking about. All right, here we go. I'm going to keep polymers and monomers up there. We're just going to switch gears. And instead of reviewing carbohydrates, let's review those lipids. I just want to click back a couple of slides. We just talked about carbohydrates. I just wanted to flash back to this picture. Remember, the individual monosaccharides are the absorbable units. Anything larger than that is not big enough. Anything larger than that is too large to cross the membrane by itself and move into a cell. It'll have to be further broken down into these individual monosaccharides. So galactose, glucose, and fructose are the ones commonly used for short-term energy storage. The other one, ribose, we're going to see in functioning for information storage in DNA and RNA. So just a reminder about that. Let's go back to the lipid review. And I'll just click past some of those. Triglycerides we started with. So let's list those other polysaccharides if we're thinking about lipids. So I'll grab a blue color. We'll now consider lipids. If we're thinking about lipid monomers, there's not many common lipid monomers. One lipid monomer that we discussed at the end of our lecture last time is this monoglyceride and fatty acid tails. These are temporary structures that exist when molecules are moving across a membrane. So I'm going to draw this glycerol backbone with 3 carbons, and then there's this single fatty acid tail. And then we have individual fatty acid tails. I'll just draw 2 columns and then label those as 2 fatty acid tails. And if we're thinking of polymers, maybe just concerning those fatty acids real quick, and we're close to lipids, I'm going to go back to this picture of a triglyceride because it shows us the chemical structure of each of those fatty acid tails. Let me switch the lights. That might be slightly easier. Fatty acids get the name because they are full of hydrogen ions, and if we start to digest these or chemically chop them up, as acids are going to do, they release hydrogen ions into solution. They increase the concentration of hydrogen ions. So 3 fatty acid tails, and I guess on that note, we made a distinction between the saturated fatty acid tails versus the unsaturated fatty acid tails. We said what they were saturated with is hydrogen ions. We couldn't fit any more hydrogen ions onto the saturated fatty acid tails. That elongated the fatty acid tails, made them tangle up more easily, and so these tended to be solid at room temperature like butter. Versus unsaturated fatty acid tails, there's at least 1 double carbon bond. So we don't have the maximum number of hydrogen bonds in this fatty acid tail. If we pulled some out of our pockets, we could stick them on there. So not completely saturated. It causes the fatty acid tail to kink up. These are smaller, more dense molecules, and so they don't stick together as easily. So it's liquid at room temperature. Triglycerides. I guess I've given that one away. We're going back to polymers of lipids. There were 3 of them that we need to know, and I've given the first one away as a triglyceride. Triglyceride is just taking these 2 fatty acid tails and sticking it onto this, what we would call a monoglyceride. Addition of 2 fatty acid tails, now we've got our triglyceride. There is the glycerol backbone. And 3 fatty acid tails I'll just draw as little columns. And now that we can recognize the structure of a triglyceride, does anybody remember the function? It's a large molecule. I likened it to a tree stump that you put on a fire and burns all night long. Long-term energy storage. So long-term energy storage is stored up as triglycerides. Not that we have to go through all this, but let's imagine you eat a big tub of Bluebell ice cream. Right? Once your cells have stored up that glucose, or say you eat a whole bag of this candy right here. Once your cells have stored up enough glucose as ATP, any additional glucose is first going to get stored up as glycogen, that polysaccharide that we had listed. But if once your liver fills up with glycogen, any additional glucose molecules are going to get stored as triglycerides. They're going to get broken down. Those carbons are going to get reassembled to build these lipids. So long-term energy is stored in this molecule, and your cells can actually take the lipids, break them down, and create glucose molecules from them. We called it gluconeogenesis. We might look at it towards the end of the semester. But if we're going to make glucose molecules from these, we're going to have to kick off a lot of hydrogens. So digesting lipids as a fuel source can shift the pH of the bloodstream. Maybe some pH scale question reviews. Questions are going to come in during our review because I know some of those are on the exam. Okay, triglycerides. We've got the structure, we've got the function. What are some other polymers of lipids? Cholesterol is another good one. Cholesterol we recognized, I guess we could have just put on the board these 4 fused carbon rings. 2 of them look like these hexagon shapes that are standing up next to one another. There's a 3rd hexagon shape that's kind of standing up, and the 4th one looks like a little pentagon shape. and it has a functional group attached to it. And we said you can rearrange the location of that functional group or swap functional groups to change this cholesterol molecule into any of the steroids. So estrogen, testosterone, progesterone, all of those molecules have their roots in cholesterol. It's the precursor molecule to all of those steroids. So we can say it makes steroids. Makes all of our steroid-based hormones. Looking at this picture that we skipped over, you can see where cholesterol hangs out. It's hanging out within this, we call this the hydrophobic moat that protects cells. It's the membrane that separates the extracellular fluid, the outside of the cell, from the inside of the cell. It's not filled with alligators, but it is filled with this double layer of non-polar tails. So you've got this hydrophobic moat. And cholesterol is a nonpolar molecule, so you see it hanging out where the other nonpolar molecules are located. We said what it's used for, to make steroids. So if we don't have enough cholesterol, we're not able to manufacture the proper amounts of estrogen or testosterone. But what happens if we have too much cholesterol? What's the bad thing there? I feel like it does, yep, it does something to the plasma membrane, and it makes something difficult about it.
Oh, it's too bulges. Because when you have like high cholesterol in your blood, it like kind of gunks up and blocks the passageway.
That's true. It can cause blockages of blood flow. But to the plasma membrane, too much cholesterol, we said it makes the plasma membrane more rigid, and it makes it difficult for things to cross the plasma membrane. So too little cholesterol is bad, and too much cholesterol is bad for membrane transport. Speaking of those membranes, what's another polymer of lipids? Phospholipids. So phospholipids we recognized as a molecule that looked something like this, where we said the head was polar, and these tails were nonpolar. And that kind of gave this molecule some dynamic properties, where if we had a whole bunch of these molecules, they would spontaneously form this double-layered membrane where one layer of phospholipids are going to face their hydrophobic tails inward, and the second layer of phospholipids are also going to arrange their tails facing the other hydrophobic tails. So we get this double layer of non-polarity. We've got a hydrophobic moat, and this is what makes up each cell's plasma membrane. So if we're thinking of a phospholipid, we can recognize the structure of a phospholipid, and we can see it's the main structural component in a plasma membrane. Different than a cell wall, these membranes we're going to see are fluid. Each of those phospholipids we will learn, I'm kind of getting into Unit 2 now, when we look more closely at the plasma membrane, we're going to see that each of those phospholipids can change positions. They can move around, they can swap whether they face the extracellular fluid or intracellular fluid. Each of the proteins can move around. So there's a lot going on there. Main structural component, we said, were those individual phospholipids. Okay. Now I feel caught up and ready to get into new stuff. Is it okay if I erase this? Everybody's good, all right. Then let's get this out of the way. We're going to keep polymers and we're going to keep monomers up, but we're switching our focus to proteins. I'm going to use, we'll try this color for proteins. So now we're looking at a protein. The monomer of a protein is going to be called an amino acid. And if we're thinking polymers of a protein, these are sometimes just referred to as a polypeptide. What is that? And if we're thinking about an amino acid, we need to be able to recognize the structure of an amino acid and how we stitch them together to make a polypeptide. Let's start by looking at the amino acid structure. We have it up here on the screen. You can see each amino acid is going to have a central carbon. And on either side, that carbon is flanked on one side by another carbon that's part of a carboxyl group. It's flanked on the other side by a nitrogen that's part of an amino group. So amino acid is where they get their name. If we look at the other sides of the carbon, it has on one side a hydrogen, and opposite the hydrogen there is what we call an R group. Maybe I can put a little box around just this one amino acid. I'll list here that there are 20 different R groups. But the other sides of each amino acid are all going to be identical. There's a nitrogen, there's a carbon, there's a hydrogen, and then one of 20 different R groups. You have in your class notes, now that we've gotten to the section of proteins, we'll see each monomer of a protein is called an amino acid. We're going to see the two functional groups. There is the amino side with the nitrogen, the carboxyl side with the carbon, and then what is unique, like I said, is the R group itself. There's 20 different R groups, and in your lecture notes you have a picture of this. It's all 20 different R groups. You don't have to memorize these. It was kind of one of the holy grails of molecular biology back in the 70s when they were trying to put together protein sequences. And it's crazy to think that we've got millions of different proteins in the body, but all of those millions of different proteins are built out of different arrangements of these 20 amino acids. Millions of proteins, just 20 amino acids. We can accomplish that by switching these amino acids around into different sequences. The analogy that's always used is the alphabet. You've got 26 letters on the alphabet, but we can make thousands of words just by switching those letters around in different arrangements. In a similar way, these amino acids are shuffled, and instead of building words, we're building proteins of different structures. I keep wanting to jump to a polymer. I'll hold off on that for a second. Luckily, I can edit that out. 20 different amino acids. What I want to say about these 20 different amino acids, now that we're still looking at the amino acid structure, we said look at each individual one. They all have a central carbon. They all have a carboxyl group on one side, amino group on the other, and an individual hydrogen. That's identical as we go from one to the next. The only difference between one amino acid and the next would then be this R group. Notice that some R groups are polar, so we would find them hanging out down here with the water. They're forming hydrogen bonds. Other amino acids have an R group that is nonpolar, so it would be hanging out up here with the other nonpolar molecules, forming the hydrophobic interactions. Then we have electrically charged R groups that are either going to be positive or negative and can form these ionic bonds. So R groups are going to be capable of forming different types of bonds, and that is going to cause the protein to fold up into different sequences. Here's where we're going to get into protein folding. There are 4 different levels of protein folding. There's going to be what we call the primary structure of a protein. It's a chain of individual amino acids. It looks like this. If we were just to get a chain, instead of me drawing the amino acid structure, I'm just going to write 2 A's to represent an amino acid. And a polypeptide is just a chain of 3 or more of these amino acids. And what I've just drawn here is what we would consider the primary structure of a protein. It's just a chain of amino acids. So the primary structure of a protein, we can see a chain of amino acids. As this chain grows longer, it's going to start to fold up into what we will call alpha helices, or these sheet-like structures. Those are then going to start to combine and form 3D shapes. And then we'll see the 4th or quaternary level of a protein. Let's start at the beginning with the primary structure of a protein. It is, like we said, a chain of individual amino acids. And we can do a little better than what we've drawn here with a chain of amino acids. I'm going to get this to-do list out of the way. We know where we're going, proteins and nucleic acids. So let's look at the protein structure a little bit more closely. And if we're thinking about the primary structure of a protein, it's a chain of individual amino acids. So let's draw a couple of those like we were doing there. Is a central carbon, a carbon to the right, a nitrogen to the left. I'll just put my hydrogens on top and then my R group below. I'm going to make this R group polar. Okay, that's fine. And I'm just going to put a box around it so we can recognize it as a single amino acid. Now let's show how these amino acids are linked together. So I'm going to draw a second amino acid, carbon to the right, nitrogen to the left. We got a hydrogen above and now an R group below. There's 20 different R groups, so I'm going to make this next one nonpolar. So these are not going to want to interact, but if it sees another nonpolar molecule, it's going to want to form a hydrogen bond with it. So nothing going on between those R groups yet, but when two amino acids get close enough, the carboxyl end of one and the amino end of the other are going to form a hydrogen bond. So I'm going to have to edit that out. Let me try this one more time. Here we are between two amino acids. We're going to see the bond that forms between the nitrogen of one amino acid and the carbon of another is a special type of covalent bond. They're sharing electrons, so this is a strong bond. It gets the special name of a peptide bond. So peptide bonds these are just a type of covalent bond. So covalent bond tells us sharing electrons. It's a really strong type of bond. What's unique about it is that this covalent bond links two amino acids, specifically between the carbon of one amino acid and the nitrogen of the next. So the next peptide bond would come if we were trying to put in another amino acid. And it would be between the nitrogen of one and the carbon of the next. So I'm just going to put an H and an R group. At the back of your lecture notes, it wants you to draw two amino acids and highlight the peptide bond that links those two amino acids. We can easily do that now. We can see we've got two peptide bonds. And a polypeptide is when there are three or more of these peptide bonds. Now we've got a protein. We've got a chain of amino acids. What starts to happen, though, as we add these amino acids together, I'm doing it right now with these markers, it also is a good chance to review something that we talked about last time with chemical reactions. There were two words that we used to describe what I'm doing right now when I'm growing a molecule, when I'm taking monomers and I'm adding them to a polymer to make it larger. What's happening when I'm synthesizing something? That's right. So what goes with synthesis? What's happening each time I add a monomer? Dehydration. That's right. The one end is donating a hydrogen, the other end is donating a hydroxide, and as we snap them together, a water molecule hits the floor. So we're dehydrating this thing in order to synthesize something. And the opposite is true if we're going to chop a larger molecule up, if we're going to decompose something. Decomposition, we said, was linked with hydrolysis. You now have to use a water molecule from somewhere. Maybe the water molecule that was on the floor, we're now using to break a hydrogen bond. Okay, so we're synthesizing these larger chains of amino acids, and here we would refer to that as a peptide bond. It's a special type of covalent bond, like we said, that links amino acids. So let's zoom out to the secondary level of a protein. The secondary structure of a protein is going to be apparent in these coils, which we'll label as alpha helices. Let's put an alpha helix, or there are these sheet-like structures that we're going to call beta sheets. These still are not yet at a functional level of a protein. A protein doesn't become functional until it takes its 3D shape, which happens at the tertiary level. So I'll say protein takes its 3D shape. What's interesting about that is that this is when it becomes functional. The secondary structure, the alpha helices and beta sheets, are due to the fact that R groups, some are polar and others are nonpolar. The ones that are polar can form hydrogen bonds between one another. When hydrogen bonds are found in addition to peptide bonds, we see the secondary structure of a protein. If we were to add the rest of the bonds, so we've got hydrophobic interactions and ionic bonds, you're then going to get the 3D shape of a protein. This is the tertiary structure. If we look inside the tertiary structure of a protein, we can see several alpha helices or beta sheets interacting together. There's one of the alpha helices, and the rest of these are those long beta sheets. At the 3D level of a protein, at the tertiary structure, we're going to see all 4 types of bonds occur. And right here in the picture, you can see the polar R groups are going to form hydrogen bonds between one another. That puts a kink in this chain of amino acids. The nonpolar amino acids are going to form those hydrophobic interactions between one another. That also puts a kink in this chain of amino acids. We've got ionic bonds forming between the cations and anions, and then even covalent bonds forming between some of these amino acids. So when all 4 types of bondings are occurring, this is where we get the tertiary structure of a protein. Now the final level, this quaternary structure of a protein, comes whenever we have 3 or more tertiary proteins interacting. So this is already by itself a 3D shaped functional protein. If we stack multiple tertiary proteins together, we can see 1, 2, 3, 4 different tertiary proteins all stacked together. This is the quaternary level of a protein. Hemoglobin would be one example. And I have a couple of other pictures of a protein, but I first want to show this short video.
Name a few of the things that they do. But maybe the most interesting thing is that these protein building blocks are themselves made up of smaller building blocks called amino acids. Here you can see a string of amino acids. The different shapes that you can see represent the atoms that make up each amino acid. And here we've highlighted 12 individual amino acids. To make it simpler, each different amino acid can be represented by a single letter.
So I don't know if you caught that. We see it this way sometimes. There were 20 different R groups. So there are 20 different possible amino acids. There are 20 different letters that are used to represent each individual R group. And what's happened here is they've used those 20 letters and they've just put them in a certain sequence to represent the sequence of amino acids. Organisms are compared at the protein level by comparing chains of amino acids. We will learn later on that each amino acid is coded for by 3 DNA nucleotides. And I'm kind of jumping the gun here, but we'll talk about mutations later on, and we'll see that at the DNA level, you can have different sequences of DNA that end up coding for the same amino acid. So there can be differences at the DNA level that don't manifest themselves at the protein level. These are going to be called silent mutations. And they actually be used to trace how old genes are. But anyway, so the protein level is a broad tooth comb way of comparing organisms. And we can now use a finer tooth comb by going all the way down to the DNA level to compare organisms. There's differences down there that I said don't manifest themselves at the protein level or at the amino acid level. Primary structure of a protein. Here's why I brought this video up, so that we can see how a primary structure folds into a secondary and then tertiary structure of functional proteins.
Now each amino acid is shown as a colored ball, looking like beads on a string. This makes the protein's structure easier to imagine. The order of amino acids is only part of the story. Because of the different shapes of the individual amino acids, they like to fold into even more interesting three-dimensional shapes.
So a bunch of alpha helices.
This molecule is twisting into several different spiral or helical shapes, and then those are folding on each other. Take a look at the three-dimensional shape as we give the protein a spin. Kind of looks like logs stacked in a fireplace.
So each of those helix structures have now been folded in a series, what look like a stack of logs, but there's a hollow chamber in the center. And this is going to be a transport protein that's going to let things like glucose move across the membrane.
Here's the one-letter amino acid code revealing the identity of each amino acid. Again, now you see the amino acids drawn to show the position of each atom. This is like looking at an atomic skeleton of each amino acid. Just like you take up more space than just your skeleton would, see how much space each atom really occupies. This is the real shape of the protein.
So very cool video there that shows how those primary structures fold into tertiary structures. I'll hold off on a second because I want to go back to our slides. We have just come off of this slide where we were talking about how proteins fold up. So the only bond that's present at the primary structure are covalent bonds, these special types of covalent bonds that link amino acids. So we will call those covalent bonds peptide bonds. So those are present at the primary level. If we were to add to these peptide bonds hydrogen bonds, then we would have secondary structures of proteins. So now we've got peptide bonds, we've got hydrogen bonds, but we don't yet have ionic bonds or hydrophobic interactions. If we add all of those types of bondings to the list, now we've got tertiary structures of proteins. Each amino acid has to be in the proper sequence if we're going to spell the right word, or in this case, build the correct shaped protein. Let me, as I display verbal mutations, come over here and finally write down the functions of proteins. We're going to learn that there's not enough space on the board for me to list all of the functions of proteins. The best thing that we can say about protein function is that function varies with structure. As you change the structure, it changes the function. Sometimes an alteration to the structure improves the function. Other alterations to the structure might impede the function, like what we're seeing here. This is an example of a normal hemoglobin molecule. It's a quaternary protein. You can see let me try that again. There are 4 subunits of a protein. Each subunit in its own right is a tertiary protein, a 3-dimensional functional protein. They have been stacked together to this quaternary level protein, and as long as each amino acid is in the correct sequence, we get subunits that are in the proper shape. But in a sickle cell red blood cell, we get hemoglobin molecules that have 1 amino acid in the wrong place. And that 1 amino acid in the wrong place causes the entire chain of amino acids to fold into a different shape. Altering the shape alters the function. In this case, it hinders the function. These sickle cell shaped red blood cells are not as efficient at carrying around oxygen and moving away carbon dioxide. Okay, so this is changing the shape based off of changing the amino acid sequence. There's another way to change the shape of a protein that does not involve changing the sequence of amino acids. That's what's unique about this word, denaturation. You can denature a protein and totally change the shape, so you completely lose the function. But we have not changed the sequence of amino acids. Right? The only bonds that we've broken here are those ionic bonds, those hydrogen bonds, and hydrophobic interactions. That chain of amino acids is still held together by those strong peptide bonds, that type of covalent bond. Denaturing a protein happens when you add heat. So cooking an egg would be one example of, as you add heat to it, the weakest bonds are going to be the first to break. So hydrogen bonds, hydrophobic interactions are the first to go. Then ionic bonds are the next to go. And the last remaining bond are these strong peptide bonds. So not only temperature or extreme radiation, but sometimes some toxic chemicals can also denature proteins. Even extreme shifts in pH can denature proteins. If you have seen any of those cooking shows where they will cook proteins with lemon juice, they're using extremely acidic pH ranges to denature proteins. You change the shape of it, it's no longer functional. This is also the threat of what happens if your core body temperature gets above 107 degrees. You ever wonder what happens if your body temperature gets too high? Your proteins in your bloodstream are going to lose their shape. They're going to denature as if they're being cooked in a pot of boiling water. And if they lose their shape, they lose their function. You've got body-wide organ system shutdown. So denaturing a protein changes the shape without changing the sequence of amino acids. The last slide about proteins just tells us if we're going to digest proteins, they have to be broken down into individual amino acids or extremely small peptide chains. Large chains of amino acids are going to have to be further broken down by enzymes. We don't have to know which enzymes break them down, but individual amino acids are absorbable units. They're small enough to move across cell membranes and into the bloodstream. Just reminding us of monomers of a protein, these amino acids. And then polymers, we don't have to know specific polymers, just that the different shapes of a protein are going to determine how it functions. Okay, take a picture of that if you want. I'm going to turn the light on and get that out of the way, and we'll switch gears to nucleic acids. Trying to go as fast as I can. I think I got just enough time to finish nucleic acids before our break time. We'll remove that. Let's use this color. We'll talk about nucleic acids. Maybe to jump directly to examples of the polymers of a nucleic acid. The two famous polymers are DNA and RNA. In this lecture, we're going to end up listing 4 differences between DNA and RNA. Maybe one way that we'll draw this is I'll tend to draw DNA in a double-stranded nature. And I stress that DNA tends to be double-stranded. It's not always double-stranded. And RNA is going to tend to be single-stranded. It's not always single-stranded. So there's one difference. If we want to look at the individual units that build DNA or RNA, now we're back to the monomers. And the monomers are going to be called nucleotides. Nucleotides. And there's 5 different nucleotides that we have found on Earth. It's interesting to think that on other planets, if there's more than 5 nucleotides, we've got a pretty small sample size of habitable planets that we've sampled so far. 5 of them we've found here on Earth. Let's look at the structure of one individual nucleotide, and I'm going to jump ahead a couple of slides. This shows you 4 of the 5 nucleotides. And since we're going to draw the nucleotide structure, just let me jump to this picture. There is the structure of one nucleotide. You can see all 5 different nucleotide bases. And what they have in common is this pentagon-shaped sugar. So I'm just going to put a generic pentagon shape and let this represent the sugar part of my nucleotide. Since everything is based around this sugar, in a chemistry class, they will label the carbons in a molecule. And hopefully you can see this in the back. It will likely be helpful for when we start synthesizing DNA molecules. This side of the sugar molecule is where you find carbon number 1. The second corner is carbon number 2. This corner of the molecule is where you find carbon number 3. Then we have carbon number 4. And then sticking out on a functional group is where you would find carbon number 5. Okay, let's build upon this sugar. At the same end as the 5 carbon on this little stalk, you're going to find a phosphate. So the phosphate end of the molecule is sometimes written as a 5' end. That's closest to the 5 carbon. Then at the 1 end, the 1 carbon has a connection where you're going to find the nitrogen-containing base. I'll just label this as the base. There are 5 potential bases. We can see all of these 5 potential bases. 3 of them have a single carbon structure. Then other ones have these 2 carbon rings that make them up. I'll just put a single carbon ring. So this will represent one of the top nitrogen-containing bases. We don't have to know the individual ones just yet. We need to know that each nucleotide has these 3 structures. It has the phosphate, it has the sugar, and it has the base. I'm going to put a box around this just to help us visualize a nucleotide. And the reason I'm doing this is because I want us to see that each nucleotide has a top and a bottom. The top side to a nucleotide is the 5' end. And down here where this 3 carbon is, this is the bottom side of a nucleotide. We call it the 3' end. The whole reason we're doing this, later on when we start to synthesize DNA molecules, we'll describe a DNA molecule as being anti-parallel. What we mean by describing this as an anti-parallel molecule, we're going to label one end. I'm going to use the red marker to label the red side of this DNA molecule. One end is going to have a 5' end on it. I'll put this as the 5' end. And the other end is going to have a 3' end to it. What the 3 and the 5' are referring to is the top and the bottom of each nucleotide. The bottom end is the 3'. The top end is the 5'. So the anti-parallel nature to these 2 strands is that if we want to label the blue one, this side is where you're going to find the 5' end, and the 3' end is on the opposite side. The other bit of information to remember is that now that we can see the monomer of a nucleotide, if we want to build polymers, if we want to grab another nucleotide and stick it onto this one, there's only one side that will accept another nucleotide, and it's the 3' end. So if we draw another nucleotide, we're going to see there is the pentagon-shaped sugar. There's going to be a phosphate on one side and a nitrogen-containing base on the other. I'll just put a single carbon ring in there. And we can label the carbons. There is that exposed 3' end that is now the bottom side of the nucleotide. You can always add to the 3' end. So if we had another nucleotide, we could put it down here. And what forms between the 5' end of one nucleotide and the 3' end of the other, I'll just use the blue color this time, this is a special type of covalent bond. It's a strong covalent bond that's going to link each amino acid. I just made a verbal mutation there. I said amino acid. That's not what we're talking about. We're talking about nucleic acids. So each subunit of a nucleic acid is a nucleotide. This nucleotide, as all nucleotides do, they have a top end. I'm just going to label as the 5' end and a bottom end that we're labeling as the 3' end. And covalent bonds form between the 5' end of one and the 3' end of the next nucleotide. We're growing now a chain of nucleotides. And you can see from the diagram on the board, you have the backbone of a RNA molecule, or these 2 backbones of a DNA molecule. This backbone here is made up of covalent bonds. So we can see how it alternates between a sugar and a phosphate, sugar and a phosphate, sugar and a phosphate. Okay, let me go back a couple of slides because we wanted to compare. We've got DNA and RNA on the list. We've drawn individual monomers those individual nucleotides. So let's compare DNA and RNA. I said we would list 4 differences between the two. And we have that one tends to be double-stranded, DNA, while the other tends to be single-stranded, and that's RNA. The second difference has to do with that pentagon-shaped sugar. In DNA, we can see one of those oxygens has been removed from a functional group. So ribose, after losing an oxygen, we will refer to as deoxyribose. So a sugar has lost an oxygen. That's the second difference. The third difference, I'm going to jump to this slide, just to reference the fact that DNA is a molecule that is confined to the nucleus of a cell, whereas RNA can move freely between the nucleus and the cytoplasm. So RNA can move freely around the cell. DNA is confined to the nucleus. That's 3. And so now our fourth and final difference between DNA and RNA has to do with those nucleotides. We can see 3 of the 5 nucleotides are going to be shared between DNA or RNA. So anytime you run across a cytosine, a guanine, or an adenine, this doesn't tell you whether you're looking at DNA or RNA. But anytime you run across a thymine nucleotide, you automatically know that you're looking at DNA. It's only found in DNA, so whether that strand is a single strand of DNA or a double strand of DNA, when you run across thymine, you know you're looking at DNA. And then uracil is a nucleotide that is only found in RNA. Sometimes RNA is double-stranded, but if you see uracil, you know that you're looking at RNA and not DNA. Okay, so we've got 4 differences between the two. The last thing that I'm reminded that we need to do is list the function of the polymers. And in the case of DNA and RNA, the function is information storage. Okay, and it makes sense. This is something that we're going to see in Unit 3. We're going to have to know all the proteins that are involved in grabbing a double-stranded piece of DNA, unzipping this double-stranded piece of DNA, stabilizing the single strands, reading the nucleotides and putting the correct nucleotide across from it, and then sealing together these fragments of DNA. There's a whole list of proteins that have their own specific role. And we'll be bogged down in learning that when we get to Unit 3. So right now, we just need to recognize when we look at the structure of DNA, what does it mean when we see a 3' end versus a 5' end? You know, we can add always to the 3' end, so this DNA molecule can get longer in this direction, where the other strand, the complementary strand, has to grow in the opposite direction. So anti-parallel nature. Since we're talking about information storage and these long DNA molecules, it might be worth mentioning that if you were to take all of the DNA that's in your cells, most of that DNA doesn't code for proteins. It doesn't build any part of you. It was originally considered junk DNA because, like we said, it doesn't have instructions for building a part of you. The segments of DNA that do build proteins that are responsible for constructing you, we refer to those segments of DNA as genes. I know we talked about this in Chapter 1. We said genes are segments of DNA that build proteins. We all have the same gene that says build 2 eyes. We did this earlier. We even have the gene that says put the eyes above the nose and beneath the eyebrows. But we don't all have the same version of that build an eyeball gene. We have, even though I'm looking around the room, I know that as individuals, our eye colors are still the same, but as a population, you can see we're experiencing fluctuation in our gene pool. Chapter 1, reviewing stuff. Before we get away from nucleotides, there is one more thing that I added, and that is, if we're thinking about polymers of DNA, there's one more molecule that we could mention, and I'm just grabbing a different color here to write this ATP molecule. It is essentially one of these nucleotides, an adenine nucleotide. We've got that sugar phosphate, sorry, I went the wrong way. We've got the sugar, this pentagon sugar, and then we've got the phosphate on one side, the nitrogen-containing base on the other. The difference is that in an ATP molecule, we've just added 2 additional phosphates. So if there's 1 phosphate, it's adenosine monophosphate. If we add 2 phosphates, it's adenosine diphosphate. And then when we add the third one, adenosine triphosphate, is what our cells are going to use as a charged up battery. So ATP is sometimes referred to as a fully charged battery. Let me move over here because I think I have just enough room to write this out. And we need to maybe mention it now because we see it a lot as we go forward. If I have an ATP molecule up here, it's a larger molecule. You've got your sugar, you've got your nitrogen-containing base, and then you've got 3 of these phosphate molecules all stuck together. And the last 2 bonds there are high energy bonds, so they're going to be holding a great deal of stored up energy. So that's why I refer to as ATP as a charged battery. Charged up battery. And what happens when you go from a charged up battery to down here, I'm going to draw an ADP molecule. Hopefully the screen's not blocking everybody out. An ADP molecule is basically the same thing plus a phosphate that we've ripped off. So we've got one of these phosphates we've ripped apart. This is a smaller molecule, and ADP is akin to a dead battery. The only one way, I think the cell phone analogy is a good way. If we think of a charged up cell phone as being ATP, it's got a full charge. We can use that charged up phone to make calls. But once we make a call, energy goes out. I'll just say energy out after we make the phone call. And now that the energy's taken out of the phone because we've made our phone calls, we're now left with a dead battery. We can't make any additional phone calls until we charge this battery up. The only way we can go from a charged battery, let me try that again. The only way we can go from a dead battery, ADP down here, to our charged up battery, represented by ATP, is we have to plug our phone up into the wall. Energy goes in to a dead battery to bring it up to full charge. Our cells don't get plugged up to a wall, but we do ingest food. So this is the chart that I was trying to explain. We've got an ATP molecule that is a fully charged battery. And anytime we remove the energy from a charged battery, now we've got a dead battery of ADP, where we get the energy to charge this dead battery up is from the food that we eat, from the glucose that's in these bags. If you ingest any of that, your cells are going to take in that glucose, they're going to rip the glucose apart, and the energy that comes from breaking the bonds in glucose are going to be used to stick a phosphate onto an ADP molecule, building the easiest puzzle that has ever existed. You got 2 pieces. You got an ADP and you got a phosphate. You stick these 2 pieces together, now we got a larger molecule. Remember that if we're synthesizing anything, this is going to require energy to go in. But if we're doing the opposite, if we're starting with something large and we're breaking down chemical bonds, then this is going to give off energy. Let me check one thing real quick. Just now that we're just about done, I can maybe share maybe too much knowledge, but I'm reminded about this fact that whenever you go from smaller pieces and you try and build something larger, even if it's the simplest puzzle in the world that just consists of 2 pieces, energy has to go into synthesizing or building something. And I guess it's this time of year, I was just talking to one of my buddies that lives in Georgia. He and his brother used to live here in Austin. They were my best, and they still are my best friends. But the younger brother died when he was 24 in 2011. So it's been 11 years. It's crazy. Actually, he died in 2010, and in 2011, we snuck his ashes into a golf course in Augusta, Georgia, where there were metal detectors and everything. But we had him in a glass vial with a cork on it, so we didn't care if we got in trouble. We got through, and then we went to the spot on the course, and me and his dad and his brother kind of spread our ashes in this one spot. And then we go back every year and kind of spend some time there if we can. But one of the years we were there in the pro shop, there was a puzzle that was a 1,000-piece puzzle. But it was of the spot on the golf course where we spread his brother's ashes. And so it was like real sentimental to me. I'm super sentimental. And I got this puzzle. And I've had it for 11 years now. And I've never put it together. Not because I don't want to. I really want to put this together and frame it and have it somewhere nice. But it just is 1,000 pieces. And it would require a lot of energy to take those 1,000 pieces and construct them to one solid puzzle. So much larger task than this, which is a simple puzzle of just 2 pieces. But anytime you're building something larger, energy has to go in. And when we're ripping apart those chemical bonds, then energy's coming out. Okay, now that I've said that, here is a slide that is maybe star-worthy because it reminds us of those 4 biomolecules that we talked about. It gives you at least one example of the monomers of each amino acids, nucleotide, monosaccharides, and at least one example of a polymer. Well, this just says polysaccharide, so you might need to remember starch, glycogen, cellulose, or chitin. For lipids, there were triglycerides, phospholipids, and cholesterol. We didn't have to know specific polymers of a protein, just that the structure of a protein determined its function. The structure was determined by the sequence of amino acids. Then in nucleic acids, they were for information storage. The 2 common polymers were DNA and RNA. And if we look at the individual units of either DNA or RNA, they are built out of nucleotides. 5 potential nucleotides. And then it's worth mentioning, since we're talking about nucleic acids, there is one of those nucleotides that has 2 additional phosphates stuck to it. And this is going to be used as an energy storage compound.